Blair McLaughlin, who is an associate researcher and adjunct professor at UC.
Well, thanks so much, and thanks for hanging in here, everyone. We're almost done. but it's Oaks, and Oaks are great. So, yeah, as everyone's been talking, I'm realizing. There we go. I've just been thinking how incredibly different it is to work with a species like this, where you have, at least 50 years to reproductive maturity. They're living hundreds of years. We don't have a seed bank. They can't survive. Acorns don't survive in the soil more than a year. So, relative to some of these annuals, so really, just as people start thinking about guidelines and things like that, how different those might need to be for different taxa. Okay, and just wanted to quickly acknowledge that these are woodlands in California. This is a map of estimates of historical lands for the different tribes in California, and you can see that most tribes have lands that overlap with oak distributions. Oaks are really culturally important species for foods, other cultural activities, and they were and still are managed often with low-intensity burn. And I'll come back a little bit if I have time at the end of my talk to this. But just to jump right in, people have been talking about this. Benefits of AGF obviously could be to help these recipient populations adapt to climate change. Another one that we haven't talked about quite as much today is the possibility of genetic rescue for threatened lineages, or particularly climate-threatened lineages, other unique lineages. And the concerns, which we've also talked about as well, so maybe we won't go into this too much right now. But so when I think of, because there are the concerns that people have brought up, when we're going to think about doing AGF for a species, what would be a good candidate species? So to me, and these are some things, but a high-value species, like something that's either culturally or ecologically important, foundational species, in my case, that has a lot of influence on the ecosystem structure and function. We want to know that it's actually vulnerable to climate change, otherwise there isn't much point in doing climate-adapted AGF. Know that it is, in fact, locally adapted to climate, and that it's unlikely to be able to track climate through means. Okay, so this is a blue oak that I'm going to be talking about today. I'm sure you've all seen them driving in and out of here, out of San Francisco, almost in any direction, except if you go into the water, you'll see them. They're just about leafing out right now, so they're like little kind of yellowy lime tints along there. They still look a little skeletal, but they are, I'd say, kind of a quintessential foundation species. They're at the bottom of the food chain. They impact nutrient cycling, water cycling, and in these systems, like if these oaks go, it pretty much turns into an invasive grassland. So that's my pitch for high value. They're also not going to track climate naturally. Like I said, they have these really long generation times, very low dispersal distances. The seeds are big. A jay's got to pick it up, carry it somewhere. It's not even swallowing it and pooping it out a while later. It's got to pick it up and put it somewhere. They're not going much more than a kilometer or so. And when you look at how where their projected habitat is supposed to be in the future, they're not going there by themselves. All right, so are they vulnerable to climate change? Well, particularly, we'll zoom in on blue oak here, but I wish I had more time to talk about this. This is a paper that I'm putting together right now that looks at, and so if you use the IUCN Red List criteria for the different categories that they have, endangered, critically endangered, vulnerable, etc., and if you use climate change projections for projections in loss of suitable habitat, this is what you get. Low estimates, high estimates. This is what they currently are. So you should be able to use climate change projections for the IUCN threat categorizations, but they're not being used right now. If you did use them, you would get a really different look at the vulnerability of a lot of these tree species in California. So we did this for all of the endemic or near endemic foundational tree species in California. And it was a big literature review on all the projections available. I won't go into it too much. If anyone wants to talk about this more, I'm really freaked out and excited about this. And for blue oak, anyways, here's our blue oak. Categorized right now as least concern. If we look at the very low estimates of climate change projections for loss in suitable habitat, and this is like if major changes are made to the way that business as usual is happening. So this is not even very likely. This is endangered. If we go along the path that we are now going and kind of going faster and faster on these days, it's critically endangered. And this is not what it should be in the future. This is what it should be listed as now. Okay, that's pitch for it. It is indeed, at least as far as the models are showing, should be categorized as pretty vulnerable. Speaking of field validations, this is some data that's going along with that, where we've reviewed all of the field studies that look at spatiotemporal changes in demography for these species. So in mortality or recruitment, the idea here being that if climate change has already started, we've got a degree of warming, we're seeing more droughts, we should start seeing in these species, say, higher mortality or lower recruitment at their xeric edges. Okay, and each of these symbols represents a different analysis, and I won't take you through the whole thing, but the red ones are, is the species responding in the way that we would expect it to if it's being impacted by climate change? And you can see that's really strongly seen in recruitment and as they relate to water or drought. Again, more on that later, but oh yeah, there's blue oak. So pretty vulnerable across the board, both in the models and in the studies that we have that are looking at what's happening on the ground. Okay, vulnerable. Sorry, I'm going really fast because I'm going to gotta get to there. Is it locally adapted to climate? So we focused in on this question of drought stress, and we did a dry down experiment in the greenhouse with blue oak seedlings from 5 different sites across its climatic distribution. The species actually has a pretty wide climatic distribution. It gets down to like about 20-25 centimeters of mean annual precipitation kind of down east and south of Bakersfield. And then it also grows all the way up in Mendocino County, and it gets like, I think, 3 or 4 times that. Okay, so we have, this was kind of a typical randomized block design, about 250 seedlings in here. Water them for 6 months and then let them dry. And we measured a lot of physiology and things like this, but this is the take-home slide that I wanted to show you, which is the source sites with their mean annual precipitation on your x-axis here. So driest source site here, this we're down by Bakersfield. Up here we're at Hopland, and you have percent seedling survival on your y-axis, and this is after about a month of having no water. Right? And so what you can see here is after that time, almost everybody from the really wet site is dead. And the ones from the very dry site got over 50% survival still. And this result holds, for those of you that are interested in greenhouse experiment challenges, it holds even when we account for seedling size. So this is not just that the dry site ones were smaller, which they actually are a little bit smaller, but it's not just that. It's not that they didn't use up all their water. So result holds even with seedling size. Okay, so it seems like this might be a really important population for assisted gene flow in a much drier future. And I say they're locally adapted to climate. We don't have the genetics information on this yet. We just have the greenhouse experiment, but it looks like they probably are. That's what I'm gonna little check. And we just got a USDA grant to do like a GWAS on those greenhouse individuals, so we can try to isolate what alleles are related to those really drought tolerant individuals. Although who knows if it'll be here in the next month, given the political situation. Okay, so I think we've got Blue Oak as a decent candidate for assisted gene flow, at least according to my criteria. And one thing I was thinking originally was like, okay, well, we should, go think about collecting some of those ones from the really dry sites. Those are really important. But here's an issue. Those really dry sites, I think that one we had is kind of down here somewhere, the one that was the really drought tolerant one in the greenhouse. This is where you're already starting to see dieback in this species distribution. So what you can see in this figure is the blue is the Blue Oak distribution, the red is the dieback, adult dieback, which that's what it looks like on the ground, but that was detected by the Forest Service aerial mortality surveys in 2015. That was after that big drought. Okay, so we've got probably the most drought adapted populations, but they're also probably the first that are going to get lost with climate change. So we can't just throw them in a sea bank, right? We can't just go collect and kind of, because acorns can't tolerate that. So what do we do? Well, one idea that we piloted here was to set up like a dispersed field gene bank that sort of doubled as a trial of assisted gene flow. And what that meant was, because it's hard to like go put a whole bunch of oak trees in one place, it's really expensive, we got land managers and ranchers and land trusts from across the distribution to kind of work together, where we collected from the areas that were in the driest parts of the distribution, and also we kind of targeted places where we'd seen a lot of die-off in that last drought. Collected acorns from there, and then the blue diamonds are the planting sites, and those are all in the wettest, or more mesic part of the distribution. And we set up little common gardens in each of those. There's, I think, 13 sites and maybe 26 little planting areas. And kind of each of those can then be stewarded by the people who are on that site. And this is all geolocated, everything's being tracked really carefully. But the idea was twofold. One, that hopefully in the future, as these populations die out in situ, we've got some of this planted in places that people can go access for restoration. And then the second question was just like, can we even will this even work, or will they all die because they're in the wrong place right now? They'll probably do really well here sometime in the future, but right now we're dealing with that climate lag issue where they've got to survive, they've got to establish and survive long enough to grow up and then be in the right place 50, 60, 70 years from now. So, let's see. Okay, so this is the first year data. Oh, and I'm sorry, I should say we paired locals and the xeric edge ones. Okay, so it's like a little miniature common garden. You have seedling survival here, local, and then the xeric transplants. So you can see you've got lower survival of the xeric ones. These are planted as acorns. And seedling height, the xeric ones are a little bit smaller. But they didn't all die right away, which seemed like a good thing. They also, the xeric ones had lower disease loads and less herbivory. So that's why I brought up that question about like maybe a trade-off. They're not growing as quickly, but they may have other things, like we saw in the greenhouse, drought stress capacities, or different types of biotic interactions where maybe these dry site ones are putting more efforts into defense compounds. And so you see this, this higher resistance to disease. Okay, and then we've been doing this for about 7 years, and they're much bigger now. You can see it over there. And last summer's data, which I literally just looked at last night. This was a great motivator. I gotta show this to some people. What happened? We had like 1 more year of funding, so I was going to do it all after that. But anyways, the seedling height differences have kind of evened out, which I thought was kind of interesting. I wasn't expecting that. And then this difference in herbivory stress looks like it's been maintained. So that I'm not sure honestly exactly what the take home here is, but I think it's that we can rescue some of these really threatened populations. They will survive, probably not as well as locals when they're dealing with these kind of time lags. And then some of these differences seem to be maintained over, at least the resistance to herbivore traits seem to be maintained over multiple years. I do have like some question of maybe, like what happened so hang on here, survival. Xeric individuals only 32% or 32% survival now, locals at 48%, so that's still a pretty big difference. And I wonder like what are the other, like 70% here of the Xeric individuals that didn't do well? Like what did we select out for them? Were they like even better at being in a dry site and they just like didn't deal with it up here? So I'm curious about what we lost, and maybe when we get start doing more genetics on these, we'll be able to tell. Okay, so these are my kind of science take-homes. Blue oaks and probably other California oaks are probably good AGF candidates. Victoria Sork is like the goddess of oak genotypes, and at one point she was like, you know what, I'm just way more worried about losing oaks than I am about losing individual lineages at this point. So, all right, that's a good stamp of approval here. I think this issue of finding populations that are at the distributional edge that might have a lot of future climate adaptive traits but are highly threatened right now is a really nice place for AGF to start. I think that seems less problematic to a lot of people because you're talking more about keeping options open, saving the totality of genetic resources that have evolved over many years. We don't want to lose those. Maybe we don't know exactly where they need to go yet, but we want them around. And then these lags between current and future climates at planting sites are really tough for these long-lived species. And it's something that I think we need to think carefully about. And then the field gene bank idea for species like oaks and other, it's a lot of these big seeded trees that are recalcitrant and can't be stored. So maybe some way to sort of extend out networks of genetic libraries where we keep the resources available for future use. Okay, I feel like I'm over it and I should probably stop there. But I'm gonna just put one little, okay, this is a tool, but I'm gonna show you that. We had a couple of, I'm not gonna go all the way into this right now, but anyone who's really interested in talking to tribes about assisted gene flow, this is really tough. I don't know if anyone's kind of started to have these conversations, but in our research group, we have been working with our tribal partners around these issues, and it ranges from like sort of neutral to like really eliciting a lot of anger and a lot of clashing of knowledge frameworks. And so I've been thinking a lot about how to do this in a way that is more culturally sensitive, and at least where we can get over initial language barriers to start talking about content. Even just words like assisted colonization, oh my God, obviously that one. But assisted migration has gotten a lot of pushback too because it recollects the idea of forced migrations of people. I know it's not what we mean when we say that, but if that's what's coming up, then it's really hard to keep the conversation going. So if anyone's interested in that, I would love to talk more about that. And a huge thanks to this is all a whole research team of our Conservation Science and Stewardship Lab. And that's it.